So our next speaker is Alex Goldberg.
Okay, so good evening. Yes, we're at the almost end of the day. So thank you very much for the organizers, and especially thank you for the audience who stayed that late. So I heard from Yael that the conference started at 8:30, and we are kind of 10 hours after that. So I guess it's a long day. So again, a special thanks also for the speakers until now. So it was very, very interesting, I would say. So because we really heard things from engineering to biology and chemistry and all around. So I will try to add something tasty or less tasty, and we'll see how it goes. And importantly, in this talk, I just want to mention that research, that project was a very emotional one for, I guess, for everybody, but also for all members of my lab. So we worked really, really actively on this since the beginning of COVID in Israel, and I think that things I will be talking also there reflect a lot of things which happened to us during that. So that's a very emotional part of the work. So we will be talking about the anti-COVID-19 specific compounds from seaweeds. So my name is Alex Goldberg. I'm from the School of Environmental Studies. And what we do, we study seaweeds. So that's a classical image from my lab. So we grow seaweeds in the sea, in the lab, in the ponds, everywhere. So almost half of the country now is covered with our systems from Eilat, I guess, to Rosh Nikra. So we tried it everywhere with the real goal to explore this very exciting marine resource, which is a marine plant, and to produce fuels, foods, materials, and other things, and especially also the new jobs. So we really pay a lot of attention about the developing of the marine environment and marine job sector. So just to show you the potential of this, here are some examples about the exploitations. Although on the right, what you see are the species which actually could be grown in Israel. So we have green algae and brown algae and red algae. But once you talk about algae, you just, people in many, many cases, but not in this audience, but think about algae as like, something like plants. So you have plants and algae. In general, these are very, very diverse species. So there are many types and subtypes. And what we did a few years ago was also to profile the chemical composition just of the monosaccharides of them. So you see them on the right there. And what I wanted to show in colors is that each of the species on the right, even though they all can be green, they have very, very different composition of even monosaccharides. So we're not getting into whole metabolomic profiling, and there are like tens of thousands and hundreds of thousands of metabolites which could be different. We're just looking into very simple compounds which are sugars, like monosaccharides, and they're also different. And if you look into the two close species like Ulvan and Cladophora, we will find that their own chemical composition of sugar is different, and there should be some expression of this diversity. So we could look at this as a source of food, but we could also look at this as a source of active compounds. And by the way, what is interesting is that all the plants you see on the right, they all grew together. So we picked them up from the 1 square meter in the Rosh Nikra plates. So they're actually growing in nature very, very, very close. So why there is a need for such a huge chemical diversity on plants which are grown in the sea, nobody knows, I guess, because if you go and sample this in the field of regular plants, you will never see this. I mean, you will have a very specific profile of compounds you will find, like especially in sugars in plants. Now, when the shutdown happened and they closed the government closed the universities and most of the labs, so we were sitting at home and thinking what actually we could be useful for, because that was very exciting, I would say, and unexpected times for all of us. And what we know for sure that seaweed have a very specific structural compounds. Specifically, they have the unique sulfated polysaccharides, which exist only in the seaweed. So that's why people actually grow them and use them. Carrageenan and agarose, all of these molecules, they exist only in the seaweed, and so these compounds are known to be active. So the exact role why seaweeds actually need these compounds is still completely known, but it is thought that since these are charged polysaccharides in the cell wall, seaweeds use them to bind nutrients or bind some other molecules in the nutrient-poor marine environment, so they serve certain as a type of the ion exchange carriers in the marine systems. That was one approach. Another approach tells that seaweed use them actually to combat the viruses and therefore because they actually are charged and they can repel things or repel cells from settling down on them. So this was what was known for us, at least, since we were not working in this area too much at that time. We also knew that seaweed-derived polysaccharides, and specifically seaweed-derived sulfated polysaccharide, have some antiviral activities. So since we were in the viral pandemic, we wanted to know what these compounds could do for viruses, and this was known already for a long time. So this was one of the first works, Goldberg in 1958, already showed that sulfated polysaccharides, specifically sulfated water-soluble polysaccharides from seaweed, can inhibit viral replication. Also, there was quite a lot of work done on different types of viruses, and there was a lot work done on HIV on these, and how the naturally derived compounds could affect them. So we actually found that these sulfated polysaccharides were tested against 28 viruses, and different types of viruses, different models. So like in general idea and the general direction was that they should be working, and that was also interesting that kappa carrageenan, one of the red algae-derived polysaccharides with the high molecular weight, was also working against coronavirus. Yes, not COVID-19, another COVID, but there was a Japanese patent we actually found, which showed that there is also a direction for the COVIDs. So at that time, what we could do, yeah. So the one thing we could do, we could grow the algae. So this is my lab in the building just nearby here. And what we had at that time was a complete indoor system for cultivation of seaweeds. So that's quite rare. There are not too many things like these globally. Usually people grow them in the ponds near the sea, but we could control precisely the medium, the environment, the lights, so in the relatively large scale, so we could produce also kilograms of this biomass. So the system we had, and we know how to grow some of the seaweeds. We also had a PhD student, Shai Schaefer, who actually was working on Ulvan, and Ulvan is a sulfated polysaccharide derived from the green algae Ulvan, and he was working on this as the active ingredient, but for plants. So what we showed and what you see here is the result of how this Ulvan looks like, and then also Shai showed that in certain concentrations, once you supplement the media of plants in Arabidopsis, you eventually could stimulate the root growth. So we got longer roots. We could actually got higher biomass yield. We got more seeds. So we did a lot of work about characterization of this polysaccharide for plant stimulation. So we actually had the protocol how to extract the ulvans. So that was exactly the time Shai was doing the experiments with Dani Haimovic's lab here in the life sciences when everything was closed, yes, and we were supposed to stop. And eventually only COVID-derived research was supposed to continue, so we were actually thinking how we can combine all of these to continue the work. And then when they came to this idea to try to use the ulvans against COVID-19 specifically. So at that time there was another problem because nobody had it. So we were in the Biological Institute and all the rest. It was very, very tough to get anybody to work with us because there was a long line of people obviously trying to test some compounds for COVID-19. So we partnered with the Southern Research Institute in the US, or it is our FDA laboratory for contaminated diseases, and they were actually very willing to collaborate and work with us on new compounds. So they exactly at this time set up the laboratory for high throughput screening of drugs against COVID-19. That was a good opportunity for them to try new molecules. So they were working mostly on small molecules, and in this case, we came to them with the large, and I'll show you in a second some characterizations with large molecular weight compounds, and they were quite intrigued about what this could do. It was a new story for them, so we could actually go and try it. So what we decided to test was the sulfated polysaccharides extracted from 3 seaweeds, because these are the ones we had in the system. So we had the Ulvan with Ulvan, we had Cladophora, also green but has a different structure and different morphology and completely different sulfated polysaccharides in it. And we have also the Hyatomorpha with another structure, polysaccharides, and it was interesting because all they are green, but for example, only Ulva has the Ulvan, which has rhamnose inside, so it's a combination of rhamnose with the glucuronic acid, and Cladophora and Hyatomorpha do not have rhamnose, but they have arabinose. We were just curious to see if these various side differences could make a change. Another thing which we actually wanted to test was if the extraction protocol effects. So we had 2 extraction protocols, oxalate precipitation and HCl, and acid extraction. So we had 3 seaweeds and 2 protocols for extracting this. Now, so these are the structures of basic structures of Ulvan. Now, what the Southern Research Institute had at that time was the cytopathic assay with the Vero E6 cells. So these are the monkey kidney cells, which expressed the EC2, to which SARS-CoV-19 usually binds. So eventually, this was the assay they already had in place, and they could screen our compounds quite fast, because these are only six compounds with the repetitions and so forth. So for us, it was very, very important actually to get to the actual virus, because many types of surrogates and so forth exist, but then they also told us that not all molecules which work on surrogates work on the actual virus. So that was a good setup for us. And so the lab actually reopened. We were able to grow the algae and purify the compounds. And so what was very exciting actually, that from the 6 compound mentioned, and we had a lot of controls there, certainly only 1 was active. So that was quite cool. So we had the Ulvan extracted with ammonium oxalate protocol, showing at a certain concentration, high concentration, and some inhibition activity for COVID, and also all the rest just didn't work. So that was quite interesting. So all the rest of the compounds which were produced either from other algae which do not have rhamnose, or extracted from the same ulva, but with the other protocols, they just were completely inactive. In this case, we got 1 active compound. So and then the next month was spent to try to characterize it, because eventually what we get with any type of extractions from seaweeds as a crude extract, so it's a mixture of many, many things. And once we started submitting the proposals, everybody wanted to know exactly what is an active ingredient. Now, there is no way we can tell now, or probably in the nearest future, what exactly the active ingredient in this mixture is, so we can go and improve and purify and so forth. But it's not it's in the immediate stages, and are not probably able to come with a single working compound. So what we do know is that we have this single algae, yes, single plant, even single thallus in this case, which was divided to 2 parts, and we extracted Ulvan with either acid extraction of ammonium oxalate extraction, as I mentioned, and 1 worked, another 1 didn't. So that was quite exciting. So we could at least compare what were the differences. So structurally, we saw that one of the working extract had a little bit different nitrogen composition, but also we did different types of chromatography separations, and we were actually able to identify several different peaks. Yeah, so this is the not working extract. So you have only peak 1, peak 2, peak 5, and this is the working extract. So we have more peaks in the middle. So at least we could suggest that there are some one or combination of the compounds which are sitting there, which might be actually working. So again, at the time we started to work with this, I personally had no idea how difficult the whole story is going to be. Yes, probably that's we were quite trying to see if anything could work. And so the more we went deep into this, the more complicated the things happened. So we did different types of separations, and indeed we just could show you that the extraction protocol significantly affected the composition of the extracted of the extract. So that was already good news, because everything in the literature, if you look today, what Ulvan is. So Ulvan is the extract from Ulvan. How it is done doesn't matter. Some papers report acid extraction, and other report just hot water extraction. All of this is Ulvan. Now, what we were able to show here, that the way you actually extract it has very, very high importance on the final activity of the compound, and also that the substructures of this changing based on the extraction protocols. Now, there were a lot of questions about the mechanism, so I can tell you that we have no idea what the mechanism is. So the review here, which came about 10 months after our work, tried to summarize the potential mechanisms of how the sulfated polysaccharides from seaweed work, because at exactly the same time there were quite a few other papers which appeared in the same area. So what we think is actually that there is a charge effects on which certain provide the shell to the cell and protect them from virus binding, but there are other mechanisms which were proposed. at the same time. So our major conclusion here, which was also relevant for many of our other studies, not necessarily related to COVID, is that the extraction protocol has a very, very significant effect on the activity of the compounds, and not everything which is called in literature Ulvan or agarose or carrageenan is actually Ulvan, agarose, and carrageenan. There we're talking about many, many different mixtures with a lot of different functionalities. And the variations which we saw were in everything: molecular weight, charge, level of nitrogen containing or contaminations or binding compounds. So there's quite a lot of complexities related to this, and the extraction protocol really heavily affects it. The weight, what we found is that ammonium oxalate-based extraction provides higher molecular weights, Ulvan, with the different charges and with a more heterogeneous composition, but also which was active against the COVID-19. So in terms of the challenges, as we identified it, we again spent quite a lot of time on trying to see how those challenges could be addressed. And a story about the active compound or compound. It can be a single molecule, can be a mixture of those molecules which work together. And so testing all of this is quite a lot of experiments to be done. The specific study was done in cells, so we definitely need other species. So, well, species, we also need animal models, and specifically for this, Southern Institute has both ferrets and non-human primates models, so it could be done there. And other issues are related to working with the natural products. So there is obviously a lot of variation based on where the seaweeds are produced and how. In our case, we could control completely the cultivation and the extraction, so we could repeat these experiments. But if you harvest algae in the wild, you could expect different effects out of them. There were a lot of IP issues about this, because once we had the materials, we wanted to work with the pharma companies, but nobody wanted to take the natural material. Yes, so it's a natural product, no IP, open to everybody. It was very difficult to get any specific corporate funding for these issues, because again, the natural, you cannot put in patents on this compound. So the issue about the large-scale use, so the issues are related to the large-scale GMP seaweed production. And we can take some examples from cannabis, how this is done today, but such things do not exist and should be addressed, or how exactly to do this, and finding the application, because one thing is to protect the cells from infection, other thing is actually to develop the application, when to use the compounds, and which way, how to deliver it, where to deliver it. So all these are questions which are still ongoing. So I just want to thank the Porter Foundation, who helped us a little bit with the seed investment in April 2020. So members of my group who came here every day when everything was logged. So Alex Shimudano was actually growing the seaweeds, and Shai and Darfur were doing the extractions, and Mike goes in for chemistry, did a lot of work with us on characterization of the compounds. Mario Lebendiger from the Hebrew University helped us a lot with the characterizations, and our colleagues at Southern Institute for Antiviral Assays and Characterizations, and Michael Lischner from Meir Medical Center, who actually did not completely discourage us, because the first thing we did when we were thinking about this was to go to physicians who were treating the first patients and asking them if this is a good idea at all to get into this, because maybe there are already treatments available. So in Meir Center, they tell us, yes, of course, go ahead. There is nothing which is working now, just do it. So there was a lot of support from their side here. And that's it. Thank you very much. I'm open for questions. Yep.
If you want to use it as a chemical in the clinic, what are the processes you have to do to identify a reliable one?
Yeah, so that's addresses the challenges. So I think one thing is to try to purify a little bit more of what we have. So I showed that we have multiple fractions here, which are different between working extract and non-working extract. So on the right is the non-working one, on the left is the working one. So we have those peaks which are different. So peak 1 and 2 and 5 are the same, but 3 and 4 are not the same. So we wanted to purify these peaks and just see if they work separately or in fractions. This is with the idea to purify and enrich the compounds. This is step 1. Step 2 is to actually grow the seaweed in the GMP environment, and so ensure that we can produce this compound, the natural compound, which will be quite replicable, Quite, I mean that we need to test both chemically and biologically and see that all the batches are showing the same result. So this is in terms of I think the next steps for the production and basic characterization. Then we need to show that those extracts, we need to find the formulation, so how we actually deliver it, because the extract is powder. Making spray or nozzles or nanoparticles in the air out of this powder is a separate question. So there are few technologies which could be explored, and eventually you can take the polymer and make aerosols out of this, which are coated or not coated, but this could be done. And then by the size of the particle, control how deep in the lungs, for example, this should go if we're talking about the delivery to patient. So after that, we need to test all the issues related to safety, yes, allergenicity, toxicity, and so forth. Then we have the animal trials, and then we probably can do the first in human trials. So these are the steps. So that's why I said that if I knew at the beginning how difficult it is going to be, we'll probably not get into this at all. But I must mention that at that time, so what we heard that it will take 3 to 5 years to develop the vaccine and 5 to 10 years to develop any drug. So that was quite a not very exciting news 2 years ago. So now the picture is a bit different. Thank you.